Welcome to Brain Ponderings. I'm the host, Mark Matson, and today's topic is very intriguing. It's one that in the area of brain research is really only taken off, at least from my understanding, a lot in the last maybe 10, 15 years. Has to do with jumping genes, and it has to do with endogenous retroviruses and retrotransposons. And so we have an expert on this, and our genome and genomes of probably definitely all animals, maybe going back to some of the first single cell eukaryotic organisms, they have acquired DNA sequences from viruses, or from, infectious agents essentially. So, and many of these are retained in the genome. And my guest is Professor Josh Dubnau. He's a professor in the Department of Neurobiology and Behavior at Stony Brook University School of Medicine on Long Island. He's has a background in uh, well, genetics somewhat, but he has had a strong interest in using animal models and particular flies, Drosophila, which it turns out has very powerful molecular biology tools for manipulating their genome. And also, actually, you can look at their behavior in quantitative ways. One can recently in an issue of Nature, there's a description of the wiring diagram of the entire fly brain. I think there's like 100,000 neurons or something, or maybe more than that. Uh, Neurons and okay, so he's an expert on recently, I guess, in particular on these retroviruses and retrotransposons, and he's using the flies both to understand how these things work, I guess basic science, but importantly for this podcast, he has evidence, others are acquiring evidence that these viral elements, if you will, play a role in neurodegenerative disorders such as ALS, Alzheimer's, and others. So welcome, Josh.
Thank you. Thank you very much. It's a pleasure to be here. I really enjoy your podcasts.
And so, uh, I looking at your website, there's a little blip that you had an early interest in insects. Now, where did you grow up?
I grew up in Washington Heights in Manhattan. And yeah, I had an early interest in insects. It started when I was a little kid, and my friend Kwame and I would play with ants. We would each get an ant from an adjacent ant hill, and we discovered that if you put them together and they were from different colonies, they would fight. And then we would, place, penny or nickel bets on whose ant was going to fight. And then because you would lose track, then we would fight over whose ant actually won. So that was an early interest. And actually, my first scientific paper was as an undergraduate. I was a middle author on a paper where we cloned the pheromone binding proteins from gypsy moths. So that really solidified my insect
Uh,
obsession. And then went on to work in fruit flies, where I've been, happily researching questions in biology for over 30 years.
and so you got your PhD at Columbia, and then you went to Cold Spring Harbor Labs. Where did you go to undergraduate? Where was
I
your
was
under?
an undergraduate at Stony Brook University, where I am now.
Oh,
And then, so there seems to be this like gravitational pull around the New York metropolitan area for me.
yeah.
So I grew up in New York City, undergraduate here at Stony Brook, went to Columbia University, where I did a PhD in Gary Struhl's lab. And that was on early embryonic patterning in Drosophila. And I actually, it's funny, I was interested to hear you trying to figure out what my training and expertise is. It's very hard to, I find it very hard to describe that because my training really is in genetics and in molecular genetics. And although I do neuroscience, I really think of myself as a geneticist.
Yeah.
A little secret, I've never taken a neuroscience course because I was in a genetics department studying embryonic development in grad school. And then I did a postdoc using genetic approaches to study memory and learning in fruit flies. That was in Tim Tully's lab at Cold Spring Harbor Lab. And then I switched to neurodegeneration over the years. And of course, I picked up a little neuroscience along the way. Yeah, so
Yeah.
where my expertise is hard to describe.
Well, yeah, but if one knows, molecular biology and cell biology, it's pretty easy to understand different types of cells because
Right.
they're mostly the same, but they, they,
Yeah,
um,
I call neurons cells, and a lot of neuroscientists get annoyed with me when I refer to them as cells, but they are cells. They're just really
yes,
unique, specialized, wonderful ones.
yes,
You-
that's right. And okay, can you give an evolutionary perspective? I didn't do a very good job. Can you give an evolutionary perspective on endogenous retroviruses and
Sure, sure.
retrotransposons?
Um, so what's interesting is that these are so-called selfish genetic elements, which means that they they have evolved to replicate themselves within our genomes. They selfishly want to make more copies. Of course, it's not quite that simple because they need us, the host, in order to survive. So if their replication went crazy, they kill cells, they kill the tissue. They quote unquote want, if you permit teleology, to replicate in the germline. Replicating in my brain doesn't help them. But if they replicate in our germlines,
Uh,
then the new copies that they make can be passed on and inherited in a Mendelian sense. They were actually, I always think it's important to say they were discovered by Barbara McClintock,
yeah.
who won a Nobel Prize for her amazing work, one of the greatest um, um, pieces of genetics ever done. And she discovered by genetics and cytology, really, that there were chunks of chromosome in maize that didn't stay in the same place. They moved from one place to another in the genome, and she correlated that with changes in the pigmentation pattern on corn.
Huh. Yeah.
So that was the origin. She actually called them controlling elements because when they moved, they changed the expression of genes where they landed. And so that was really the origin of them in terms of the history of their discovery.
And Josh, and so then, no, the corn, the sweet corn we get, they're all yellow, but we used to call it Indian corn,
Yeah,
right? With,
yeah, that's the
now
kind of
that's due to the jumping genes?
yeah. So that beautiful mottled pigmentation pattern, both across and within um, kernels on the ear of corn, is caused by this dissociator element that she discovered that moves around the genome. Now, it turns out that transposons come in several different flavors. There's the type that Barbara McClintock discovered, which are so-called DNA elements or type 2 elements, we call them now. They're not believed to exist in fully functional form in humans. And they're not the type that my lab studies, but historically they're very important and they're very important evolutionarily and in biological systems. They basically cut themselves out of the genome and they paste into a new location. They literally, like, they have a recombinase that has, like scissors almost, a protein that recognizes the ends of the element that actually codes for the protein that's doing this. They clip it out of the DNA repairs, and then they cut DNA somewhere else and insert themselves in. That's the so-called cut and paste. The type that I study and the type that a growing number of us believe may be very important for human health and disease are so-called copy and paste. They also exist in our DNA. They sit there as a length of DNA in our chromosomes, and they code for an RNA that codes for one or more proteins that binds back to that RNA and makes a cDNA copy that it inserts back into the chromosome at a different location. So they're called retrotransposons, and they're evolutionarily analogous to a retrovirus. Everyone's probably heard of HIV, right? So HIV, when it infects cells, it inserts a provirus into the DNA of that cell, typically happening somatically, not in the germline.
Right.
And that can sit there latently or it can become activated by some cellular stresses or by, things that are not fully understood. And it goes through an RNA intermediate, like a retrotransposon, makes a cDNA copy, goes back into the nucleus, and inserts back into a new location. And what HIV does is that replication cycle is between cells. So it's packaged into the RNA copy is packaged into a virus that moves to a new cell, fuses with that new cell, and then starts that cDNA copy goes into the nucleus. Whereas retrotransposons do that all within a cell. And so my understanding is that evolutionary virologists think that the evolutionary ancestors of all retroviruses are retrotransposons. That first there were retrotransposons that then picked up the ability to package into a virus and leave the cell with a surface protein that binds a receptor on a neighboring cell, a different cell, and then integrates into that cell's genome and repeats that infectious cycle. But what's interesting is they've probably gone back and forth over evolutionary time. Retrotransposons evolved the ability to leave the cell, picked up this envelope protein that lets them fuse to a new cell, and became a retrovirus that could then become infectious, move from one individual of a species to another, and then get into the germline by mistake, and then get inherited, and then over time lose the ability to leave the cell and become a retrotransposon again. And then there's ones that we call endogenous retroviruses that kind of straddle that continuum. They look like retroviruses. They have all the machinery of a retrovirus. They look like they should be able to move between cells, but they're in our germline and we pass them on in a Mendelian way. And they could either be on their way out to become a retrovirus or on their way in to lose the ability to be infectious and become a retrotransposon. And that's actually the type that my lab has been most interested in the context of brain and neurodegeneration.
Yeah, because your point about their ability to go between cells is very important because the pathology, and whether it's Alzheimer's, Parkinson's, ALS, frontotemporal dementia, there's these accumulation of proteins that seems to be start in one area of the brain or spinal cord and then move between neurons. And so that's really what you've been working on a lot, right?
Yeah, that's something that really we've become fascinated with. What's interesting is, I'm not a so-called prion biologist, but this idea comes from this field of science that studies what are called prions. So everyone's heard of mad cow disease, and similar infectious neurodegenerative disorders. These are ones where you have cows that are sick, and then if you get that into the food chain and we eat that tainted meat, we can get a neurodegenerative disease that is infectious in that way. And brilliant work so there are these prion diseases that are infectious, and brilliant work by many people, including Prusner, who won a Nobel Prize for this work, demonstrated these are infectious by a proteinaceous infectious agent. And the way they are infectious is so proteins fold in a very special way in order to do their job. And these have the ability to undergo like a conformational transition into a new shape that is pathological. And when a protein has undergone, a prion protein has undergone that transition and bumps into another protein of the same sequence, it can induce it to undergo that conformational change. So that's like a replication event of sorts. And it's thought that these can move between cells. So it was discovered that many of the proteins that pathologists have found to be implicated in Alzheimer's and ALS, in frontotemporal dementia, in a whole suite of neurodegenerative disorders, that many of these proteins that pathologists see accumulating with mislocalized in the cell or outside the cell in the brains of people who died of these disorders, these proteins share some biophysical features of the prion protein. They have domains that are reminiscent of prion proteins. Domain is like a part of the protein, a region of the protein that has some string of amino acids that has some structural and biophysical properties that are similar to these prion proteins. And then it was discovered that indeed they can aggregate by maybe a similar mechanism and that they can seed that transition when they bump into another protein of the same sequence. And indeed, they can move between cells. So that was like an aha moment when people studying the field thought maybe this mechanism explains why, when somebody is diagnosed with ALS, it might be focal, they might notice problems in their right hand. And then over time, it moves along the length of that arm and spreads to the other side of the body and spreads as if it's moving along the topographic organization of the motor system.
Yeah.
And so people thought, well, maybe it's moving between cells. And then people have been able to show that in culture or in mouse models and in fly models, that these proteins actually can move between cells. So we thought, a lot of people thought maybe that explains this, what a cell biologist would call non-cell autonomy. That's a word that basically means something happening in cell number one can influence what happens in cell number two. It's a very intriguing idea, and there's some evidence for it, quite a bit of evidence for it. But ALS and frontotemporal dementia are not like prion diseases. They're not infectious. You don't get them by eating tainted meat or by living with people that have the disorder, or there's no evidence for that. And in animal models, so with the prion disease, if you purify the infectious form of the protein from one brain of an infected animal and you inject it into a mouse, from one mouse into the next, the recipient mouse gets sick. Yeah. And it dies with a latency that is very stereotyped. And then you can serially passage that. So it follows what's called Koch's principles of an infectious agent. That's not true for the types of proteins that are involved in Alzheimer's disease and ALS. It's never been found to be true. Part of it is true. If you inject misfolded protein of, proteins like TDP43, the one that I principally study that's implicated in ALS and frontotemporal dementia and to some degree in Alzheimer's, if you inject that misfolded protein into a mouse's brain, you can see the protein moves, but it doesn't cause disease.
Yeah.
So there's something missing, and we're investigating the idea that that something might be these endogenous retroviruses. What we believe is that there's a cycle, a feedback cycle. We've shown that if you disrupt the protein by having it aggregate, this TDP43 protein, having it misfold and aggregate by its prion-like process, that causes the endogenous retroviruses in that cell to increase in their expression. And similarly, if you just increase the expression of the endogenous viruses, it triggers the protein to aggregate, mislocalized to the cytoplasm. So it's like a cycle, and that cycle runs between cells as well. So, yeah, go ahead.
So maybe a less specific type of cell stress also induce reactivate. In other words,
Yeah.
So, we did all this work early on suggesting that the amyloid beta peptide, when it's aggregating on membranes, causes oxidative stress and lipid
Yeah.
peroxidation. Um,
It's a wonderful question. And I should say that I should just back up for a minute and say I want to give credit to a growing field. My own lab is focused on TDP43 in flies. Others are studying the same connection of retrotransposons of various types, including endogenous retroviruses with TDP43. And that includes, Eddie Lee and Molly Hamill and, a number of others. Roger Scher here at Stony Brook. And then there's a whole other group of people who are studying similar ideas in the context of Alzheimer's, in particular with tau, so Bess Frost and people like that. But your question was about upstream stressors, because
Right.
that's really a key question, is that we all wonder whether or not you believe in this retrotransposon idea or not. Most of us who end up getting a neurodegenerative disease it's not predicted by our genetics. It's really
Right.
predicted just by age.
Right.
The risk of Alzheimer's or ALS or frontotemporal dementia just increases with age. So me being 58 means that in the next 20 or 30 years, I have a higher risk of some neurodegenerative disease onset, even though nobody in my family has,
Mm.
there's no strong family history of that, right? And so, what is it that triggers that? Why do some people get it and some people don't? And so, the whole field thinks about what's that initiator, what or initiators, and I think we still don't know. Um, but one thing is really, really a strong theme within the transposon field is that they're activated by stress.
Yeah.
There's like wonderful work in plants that shows that like when the salinity of the soil increases, transposons start jumping. Anything that really stresses the organism. Um, if you want to put some teleology on it, a transposon that might say to itself, the environment in this cell is not so good. Time to get out. At least for the ones that have the ability to move between cells.
Has anyone looked yet? I haven't really followed this area much, but as you know, we did a lot of work relevant to brain aging and neurodegenerative disorders, and we got interested in how to reduce risk by lifestyle modifications. And one thing we know counteracts aging is reducing energy intake, calorie intake, or intermittent fasting, exercise. So, has anyone done in like mice look at these? Look in old animals that have either have or have not been on calorie restriction for a long
Yeah,
time.
it's a great question. So there's work in mice on retro just talking about aging, not even neurodegeneration, right?
Right.
Age dependent
Right.
decline. There's work from the Gorbanova lab in collaboration with John Sedevi in mice, looking at the increase of retrotransposons with age, um, and even looking at inhibitors of retrotransposons as life as extenders of life. There's work from Steve Helfand's lab in flies actually showing that retrotransposons increase. He was looking mostly in fat, which is also has sort of like a liver function in flies, that the retrotransposon levels go up with age, and that if you do caloric restriction, which extends lifespan, that you delay that onset.
Yeah.
So that I thought was really cool. We've shown in flies back in 2013 that in the brain, retrotransposons actually begin to replicate with age. They actually, even in non-dividing neurons, they appear to be able to make new copies of themselves, which can't be good. I mean, you
Yeah.
don't want genomic instability in a cell that in cells that can't be replaced by, effectively by new division. So caloric restriction has been found to have impacts. And then my lab has now gotten very interested in what I would call psychological stressors. And I put it in quotes because we're using Drosophila. So whether you'll permit me to say psychology, but
Yeah.
there's epidemiological evidence that loneliness, PTSD, anxiety, depression are correlated with increased risk of neurodegeneration in humans. It's very hard to prove causality because, is depression a prodrome, of neurodegeneration? Are the medications that a person who has, anxiety, depression takes, are they predisposing? It's very hard to
Mhm.
show causality. So we're trying to play around with that in flies, and we have found that early life social isolation uh,
Okay.
increases the rate of neurodegeneration in genetic models that that are predisposed to it. So what we mean is we first from the first moment that the adult fly comes out of its little pupal case, we separate them. They're all alone, and that's very stressful to them. Others have shown that it causes loss of sleep and lots of stress. If we do that for a period in early life, then group them again, it's too late. When
Ah,
we group them, we induce this transgene that triggers neurodegeneration, and we look at the rate of that. It increases the isolation increases the rate of neurodegeneration and even increases the rate of spread between cells. We don't know why yet.
interesting.
So we're playing with things like that,
What we're getting
stressors. Yeah.
that's Yeah, now you got me thinking about a lot of other things. I don't want to get too much off topic, but right. So there's this paradox in mice and rats and maybe in humans where chronic psychosocial stress elevates cortisol levels, right? You get kind of chronic activation of that stress endocrine, neuroendocrine stress pathway. But caloric restriction and intermittent fasting also elevate
Yeah,
cortisol levels.
yeah.
And we'd found, and it's kind of got buried and people haven't picked up on it. We didn't either, and I think it was the year 2000 or 2002, we published a paper where we showed, in contrast to bad stress, the good stress, if you will, of caloric restriction um, has a different effect on the receptors
Hmm.
for cortisol. There's two receptors, the glucocorticoid receptor and the mineralocorticoid receptor. Bad stress decreases MR and GR stays up, and caloric restriction does the opposite.
Interesting.
So even
I'll
though
look
the
at that paper.
cortisol,
Yeah.
even though the cortisol levels are up with caloric restriction, the way the cells are responding is different.
Yeah. So there must be some other aspect of the stress in addition to cortisol.
Okay, so
Yeah.
so in Drosophila, my understanding is they don't have adrenal glands,
They don't have, yeah,
and they don't.
exactly. Right. And so we don't know yet. So the big question, this is early days for these kinds of playing with psychological stressors. But yeah, the next thing to do is really to understand what are the mechanisms of that.
Yeah, because there
Yeah,
could be neurotrophic factors, right? With
could be.
depression, BDNF is down, and so on, and there could be fly trophic. There's undoubtedly are fly trophic factors, and
Yeah,
so
yeah, yeah. We don't know. We don't know what those. It also could be. It's known that flies lose sleep when they're isolated, so it could be just chronic loss of sleep.
uh,
Very hard to know at this stage.
yeah.
Yeah, yeah,
Could you start then on your journey to, from your basic genetics and the fly uh, and so on, looking at development to now being in the neurodegenerative disorder field. What were kind of your key studies in the flies?
yeah,
How did you approach this over
yeah.
time?
Thanks for that question. I mean, I often compare myself to my dog. I go for a walk with the dog and it's sniffing, and then a squirrel runs across its path and it's just like, you know, and that happens to us as scientists sometimes.
Yeah.
You're studying something, you get some odd result, and you kind of think about it, and you have to decide, are you going to be sidetracked by that? So for me, I mean, I transitioned from early embryonic development to memory from grad school to doing my postdoc just because I saw an amazing talk by Tim Tully, and I thought that's completely uncharted territory, which it really felt like at the time, and I moved to that field. And then when I started my assistant professor position at Cold Spring Harbor Lab, I was studying memory and learning, and a grad student in my lab, Wan-Ho Lee, who's now an assistant professor at Texas A&M, she discovered that one of the memory mutants that came from a genetic screen to find mutations that impacted the fly's ability to retain memories was a small RNA, a non-coding RNA called a microRNA. And she studied its role, and it turns out it regulates a dopamine receptor. But she, just on the side, results that we never published, was trying to find the genes that this microRNA regulated, and she found that it was predicted to regulate from computational analysis from looking at the sequence many of the same genes that another protein called pumilio regulates. And pumilio is an RNA binding protein that also was involved in memory. So I thought, well, maybe they're interacting. Let me start manipulating this class of proteins called argonauts that are the ones that co-regulate with the small RNA. Because I thought maybe the argonaut proteins and the pumilio protein and the small RNA all come together to regulate the translation of the same RNAs. And I started doing genetic interaction screens in my own office. The Drosophila faculty often keep flies in their offices. I've stopped over the years, but I did for many years. And I discovered an interaction between pumilio and an Argonaut protein of the wrong type. It was an Argonaut protein that regulates transposons. And that was just really odd. And I started thinking about it, and Wan He Lee, the grad student in the lab, and Lisa Prazak, a postdoc in the lab at the time, started thinking about it with me. And we just said to ourselves, either this Argonaut 2 protein that regulates transposons is doing something else for memory, or maybe transposons are doing something.
Yeah.
Now, Rusty Gage, I have to give credit to Rusty Gage and his colleagues. They'd recently published this idea that retrotransposons could replicate in mouse brain and in human brain, and others, Chris Walsh and Jeff Faulkner had also published that sort of stuff around this time. So we were primed to think about transposons in the brain. And then over the years, I just, like a dog following a squirrel, I started studying that, and then it just took over my lab. Yeah, like an
Yeah, and
infection.
you're well on your way to catching the squirrel and
I hope so. Yeah. Yeah. I mean, what's exciting to me is that there are several clinical trials now um, that all focus on this idea, testing reverse transcriptase inhibitors. These are drugs that were first developed for HIV/AIDS, and because HIV, like retrotransposons, has to make a cDNA copy from the RNA in order to complete its life cycle. And there's really good drugs that stop HIV from doing that. Now, they're not great at getting into the brain. We don't know if they'll work as well against retrotransposons, depending on the drug class, but there are trials now to test that. And those are exciting.
and are they specifically inhibit the reverse transcriptase. I mean, there's reverse transcriptase that play normal function. Telomerase comes to mind.
Yeah. So
But
it's a great question. The early classes of nucleoside analog reverse transcriptase inhibitors probably were not specific enough, but my understanding is that the newer generations are really quite specific for reverse transcriptase
interesting.
and don't seem to hit telomerase that much. But no drug is going to be
And
without
they,
it.
they get into the central nervous system across the blood.
There are, there are companies working on this now. There are
Yeah.
ones that get into the CNS pretty well, and I think there's some focus on those. Of course, every time we design a drug intervention, we're making a set of assumptions. Even if the retrotransposon storm hypothesis, as we call it, turns out to be correct, it doesn't necessarily have to involve replication. So
Yeah.
these drugs make the assumption that copying the RNA into DNA is critical to the toxicity of these elements. Could just be that their proteins or RNAs are toxic. In fact, there's evidence that they can look like a virus to the cell, and that either their RNA or their cDNA that accumulates in a cell can activate different pathways that tell the cell you've got a viral infection, and it could lead to sort of chronic
Yeah,
inflammation,
Yeah.
cytokine, storm kind of stuff.
Yeah, and there are elegant mechanisms that cells have evolved to deal with that. We did a little work on what are called toll-like receptors
Yep.
and had some evidence they have normal functions that seem to be independent of any role in recognizing infectious agents, which kind of makes sense to me. I mean, why would you have these proteins like toll-like receptors sitting around for a lifetime, maybe not ever being exposed to a bacterial
Yeah.
or viral infection? So,
Yeah. I mean, evolution tinkers. So there's a great quote from Jeff Hall, a great geneticist, Drosophila geneticist,
yeah,
who studied circadian rhythms and shared the Nobel
yeah.
Prize for that. He said, "We're all studying the same genes, we just don't know it yet." And he was thinking of that as like a so-called forward geneticist. We discover genes that impact our biology, but
Yeah,
because there's no gene for this or that. They're
right,
all what we
right.
call pleiotropic. They
Yeah,
do many things. You and I studying different areas of biology could end up studying the same gene without realizing it. Yeah,
yeah.
yeah.
Okay, so then continue with the story. So
Uh,
how did you
yeah.
you're doing a lot of work on this TDP-43 now.
Yeah. And okay, so we started thinking about transposable elements, retrotransposons.
Yeah.
We discovered first that in normal aging, that they just increase in expression in the brain. And that was a 2013. That was really the first paper where we, established this sort of connection to the brain.
Yeah.
And then I started thinking about TDP-43 because so I started thinking about neurodegeneration because we saw this when we unleashed transposons by knocking out one of the control genes, an Argonaut protein, the control gene, we saw very rapid age-dependent memory impairment, and it turned out to be like accelerated neurophysiological decline. It wasn't specific to memory. So I started thinking about neurodegeneration. And when I started reading the literature, TDP-43 caught my eye because its unstructured domain that is implicated in its prion-like conformational change looked a little bit like a class of proteins called GW proteins that bind the Argonauts. Now, that may turn out to be a red herring, but that's what I saw looking at protein prediction algorithms. And so I started thinking maybe TDP-43 is in here. And so we started studying TDP-43. And
So
as
TDP-40, yeah, it is very interesting. In some ways, it's different. Well, of course, it's different than tau or alpha-synuclein. But what's interesting to me is my understanding, it's normally in the nucleus,
That's right.
right? And it can bind to DNA, it can bind to RNA, but this protein, in addition to having these regions, you can bind to DNR. It can move out of the nucleus. It can move into, there's like a nuclear localization signal, a nuclear export signal.
Exactly. Yeah.
So it is moving around between nucleus and cytoplasm, whereas tau, as far as we know, it's mainly in the cytoplasm.
Right. And
It,
that's right. I mean, TDP-43 shuttles back and forth. If you just look with antibodies to visualize TDP-43 in tissues or in cell culture, it looks nuclear because the majority of it is in the nucleus at any given time in a healthy cell. But yes, it shuttles back and forth, and it has, as you say, this nuclear exit motif that lets it leave the nucleus, and then a nuclear localization signal to go back. And but what pathologists see in brain tissue from affected individuals, or if they look in the right regions of the brain, they see that many of the cells, it's sort of lost from the nucleus, and it just accumulates in these big, what are called inclusions, many, many copies of the protein clumped together in the cytoplasm. Probably that acts like a sink, so that when protein is in the nucleus and exits, it accumulates there. Although I should say there's some evidence that aggregation begins in the nucleus. We see this in cell culture. Whether that's really what's happening in the patient in vivo, we don't know. But it looks to us like the sort of pathological misfolding begins in the nucleus and then moves out into the cytoplasm and then sort of accumulates in big aggregates.
Now,
But
what's the
yeah,
link of this, Josh? So I did two podcasts with David Rubenstein on autophagy, right? And there's a lot of evidence that the ability of, well, certainly the ability of cells to clear out these misfolded self-aggregating proteins is compromised, but there's quite a bit of evidence that the autophagy machinery itself is compromised. So do these uh, retroviruses, retrotransposons, they increase with age, and do they affect autophagy?
it's a good question whether they in themselves directly affect autophagy. I don't know the answer to that. And it would be interesting to look if there's any direct literature on that in, let's say, cell culture. What's hard here is that we have these feedbacks because
Yeah.
the protein aggregates triggers endogenous retroviruses. The endogenous retroviruses trigger protein aggregate. So saying which came first can be a little bit hard. And then autophagy certainly declines with age. I think a really interesting question is, why are these diseases of age? I mean, we don't get, ALS when we're 10
No.
or Alzheimer's when we're 10 or 20. Um, Presumably, they're all of these biological processes that change with age
Hmm.
that are sort of colluding to increase risk.
Yeah.
But it really does feel like a multi-hit thing.
Yeah.
Like you have this background of aging, and then something can happen. And if that something happens when you're 65, it's more likely to trigger some self-sustaining process than if it happens when you're 25. And if it happens when you're 75, it might be even more likely. Um, yeah, not a fun thing to talk about at the age of 58, but
Well, I'm a decade older. I'm 10 years older than you. So,
and I mean, some of us just are lucky and never get these kinds of disorders. So knock on wood. Yeah,
yeah. Yeah.
yeah,
All right. And then, okay. So why flies? So it would be good. I don't know if I don't think I haven't had a podcast with a fly person. So
yeah,
could you explain well, one advantage is you can keep thousands of them in your office
yeah,
and look at their,
yeah,
quantify the, but from the molecular genetics and manipulating, the, in this case, neurons in their brain.
yeah. Yeah, I love the why flies thing because I mean, we only have, you know, I could go for hours. No,
I
I'm
know,
joking.
I know.
Um, one great reason for the vertebrate researchers out there is no Cook. We,
Yeah.
we don't have to do animal protocols. Nobody cares if we're mean to flies. Um, no, fruit flies are just this incredibly powerful system for genetics. So the whole genome is sequenced, as is true for many species. The whole brain is mapped. So we know every neuron and its connections, every glial cell. Um, there's this Lego-like toolkit where you can choose from tool one and tool two and put them together. So just to give you an example, there are collections of thousands and thousands of fruit fly strains that exist in a stock center in Bloomington, Indiana. Mutations in any gene I want, for the most part, or transgenes that are called RNAi that basically target a gene to knock it down. And each of these reagents is made in such a way where it's turned on and off by a yeast protein called Gal4.
Yeah.
And then there's a whole panel of strains that respond to Gal4. They will turn on if the cell has Gal4, literally thousands of strains, tens of thousands. Then there's another panel of strains in which Gal4 is expressed in any cell type you like. So I could say I would like to express gene number one in astrocytes, or I'd like to knock down gene number one in astrocytes. And I just have to choose strain 237 from here and strain 1592 from there, take a male from one and a female from the other, put them in a vial together, and then 2 weeks later, I have a population of flies where I've knocked down or overexpressed that gene in astrocytes. And I can even add a third trick, which is called Gal80, a temperature-sensitive Gal80
Hmm.
that converts the Gal4 into something that can be turned on and off. So now I can say, I want to knock down or overexpress a particular gene only in astrocytes, only at the end of development, and only when the flies are 10 days old. And I want to turn it on or off for 6 days. That's possible. Um, so it's just this incredibly powerful system. And then their lifespan is shorter. So, I joke that when we do these longevity experiments where we have lots of genetic groups and manipulations and we're measuring how long the flies live, it means that a student or a postdoc in my lab or a technician has to take all of those vials of flies, transfer them to a new vial every other day, count how many died, and continue the experiment until they're all gone. And so it might take 80 days until they're all gone in a fly. So when a reviewer says, do a new experiment, we go, oh, it's going to take 80 days. But in a mouse, it's years if you want to go until they're all dead.
Yeah.
It would be several years. And we can have, 180 flies per group in a lifespan experiment,
Yeah.
all sitting in a rack in an incubator.
Yeah.
So there's an economy of scale.
Yeah.
On the other hand, they're evolutionarily more distant from humans. And so there are questions that you can't ask in a fly. If you're really interested in like cortical circuitry, they don't have a they don't have a cortex. They don't have a
Yeah.
spinal cord. So if you really want to study this, why, the questions that are very specific to the motor neuron, lower motor neurons in the spinal cord, you can't do that in a fly.
Yeah.
I treat the neurons as test tubes to study cell biology.
Yeah.
And they have all the same neurotransmitters for the most part. They have GABA and they have acetylcholine and they have glutamate and they've
Yeah.
got lots of analogous neuropeptides. So you can study those cell types, dopamine, but if you really want to study the circuitry that's specific to a human, you're not going to find that in a fly, and you might find it in a mouse, or you might not.
Yeah. All right. And uh, let's see. So continue along the line. So, and can you introduce introduce retroviruses, retrotransposons, or how do you manipulate specific
Yeah. So
sequences,
we, it's a great question. So yes, we can, these retrotransposons or endogenous retroviruses, they have their genome,
Right.
which sits in our genome, is usually about 7,000 bases, something like that. We can introduce engineered transgenic copies of retrotransposons or endogenous retroviruses. So we can turn them on and off,
Yeah.
the same tools that I just described. We can engineer them so that they're tagged so that when they replicate, they'll turn on a fluorescent protein. So then we can look in the brain and go, oh, the neurons or glial cells that are glowing red, those are the ones that had a replication event. And
Yeah.
then we can look in those and go, oh, look, they have aggregating TDP43 and they have DNA damage. We can look with markers that say, is the DNA, are the chromosomes broken?
And you're introducing the human gene encoding TDP.
No, it's been suggested that that might be an interesting thing to do, to take the human endogenous
You're doing the
retroviruses.
fly
But
homolog?
we're doing the fly. I like to say analog because it's not exactly
No.
clear that this endogenous
Okay.
retrovirus that people have called it gypsy for many years. There's now a new name, MDG4. So we call it MDG4.
Okay.
It's a fly analog, I like to say,
Okay.
of human endogenous retrovirus K. They're very similar in structure and function, but they're not like, it's hard to say this one is the same as that one in humans because
Yeah.
the suite of retrotransposons is a little different.
All right. And how did you establish? So you had several nice papers over the last few years, and one of them you provide pretty strong evidence that of cell-to-cell transfer mediated by the retrotransposon.
Yeah. So that was done. So in that paper, uh, we manipulated both the fly and the human endogenous retrovirus. The fly one we manipulated in flies and in fly cell culture. The human one we only manipulated in human cell culture. With both of them, we showed that if you introduce an extra copy of the retrotransposon, endogenous retro, I sometimes say retrotransposon, sometimes say endogenous retrovirus, same thing. If we introduce a copy and overexpress it, that is sufficient to trigger aggregation of the TDP43 protein. It leaves the nucleus and it clumps in the cytoplasm in both fly cells and in human cells. In fly cells, we're able to study a tagged version of the endogenous retrovirus that expresses this fluorescent protein. And we used what's called a transwell system. We had tissue culture growing on two sides of a membrane, and the membrane had a pore size that was too small to let cells move, but was big enough to let viral particles move. And when we introduced the virus, the endogenous retrovirus, an extra copy of it, and activated it in cells on one side of the membrane, and then we imaged the TDP43 in cells on the other side of the membrane, the TDP-43 aggregated across the membrane, but especially in the cells that that showed that red fluorescent protein that said they had received the virus. And if you delete that envelope glycoprotein, that is the surface protein of the virus that mediates its fusion to a new cell, it block the ability to cause TDP-43 aggregation on the other side of the membrane. And there's a wonderful paper from colleagues in Germany who did something similar with tau, but not using the fly endogenous retrovirus. They showed in their experiments that aggregated tau, misfolded tau, was physically moved in the virus from cell to cell using
Huh.
mammalian endogenous and exogenous viruses.
Huh.
Yeah. So,
So what?
yeah,
So the tau is binding to to a viral protein like uh, you mentioned glycoprotein that which is like viral coat protein, right?
coat protein. Yeah. What they found, I don't think they know anything about the biophysics of, what is it binding to,
Yeah.
but they were able to show that aggregated tau is transferred in these. They're basically they may not be fully functional viruses. They're sort of like extracellular vesicles that have this
Uh,
end protein, the glycoprotein on the surface. So it may not require that the virus be fully functional in order to transfer tau, but it greatly, there are these experiments where people have cells that have so-called strains of aggregated tau, and in those, the ability of those to trigger um, movement and aggregation of tau in a recipient cell was greatly enhanced in the presence of these viruses.
yeah, interesting.
Yeah.
You mentioned the extracellular vesicles. That I had a podcast with Dimitros Kapagiannis, who was a neurologist in my lab, and he did a lot of actually some of the first work showing that he developed a method you can from the plasma of humans, of mice, rats, you can first isolate all these extracellular vesicles. There's commercially available kits for that. And then he used the antibody against a neuronal surface protein, L1, to pull out the vesicles that are probably coming from neurons. And then he looked at tau and A beta and so on in AD patients and had some nice papers. But uh, we, we and others have evidence that these vesicles may be a way of transmitting pathogenic proteins from one cell to another. And my own idea is that the cell where it's starting to accumulate wants to get rid of it, right? Because it's a mechanism,
Yeah.
you got this pathogenic protein, it's causing problems inside me. I'll just, package it in these membrane vesicles and spit it out and let some other cell deal with it.
Yeah. That's a really interesting perspective on it. It's not it's almost like we've been touching different parts of the elephant, but we're starting to come together here because this sort of prion-like spread idea and this endogenous virus spread idea are not incompatible with each other. They
Yeah,
could be coordinating. The
yeah,
other part of the literature that I feel is coming together with this is this recent literature on ARC. So ARC is this
yeah.
activity
Synaptic
regulated.
plasticity protein.
Yeah, it's this
Yeah,
famous plasticity protein
yeah,
that's been found to be involved in, synaptic plasticity,
yeah.
underlying memory and learning. And independently, groups studying the fly version of ARC and the mammalian version of ARC discovered that it's actually an ancient retroviral gag protein, and that it actually
Hmm.
transfers between cells in the nervous system bound to its own RNA,
Huh.
and that the RNA may get translated in the recipient cell. So it may be that this ancient viral property that was infectious and not good for us has been co-opted independently in insects and in mammals as an intercellular communication system in the nervous system but unfortunately, it may also contribute to the propagation of these prion-like misfolded proteins between cells. That's where my head is right now. Obviously, that's a lot of supposition,
That's
but yeah,
really intriguing, right? So the idea then is that ARC, this protein that's critical for synaptic plasticity, learning, and memory, uh, is, so we should be very grateful that these viruses infected cells early on, because otherwise we wouldn't be able to learn and remember anything.
it could be. I mean, there's such a wonderful literature on this. My favorite one in some ways is that placental mammals have this syncytium. The trophoblast is a
Yeah,
syncytium where cells are fused.
yeah,
Apparently, the what gives the cells that fusogenic ability is an ancient envelope protein, this envelope glycoprotein that allows viruses to fuse, retroviruses to fuse with a recipient cell. And so we, as placental mammals, have co-opted in humans, it's HERV, human endogenous retrovirus W, which is now a defunct virus. I mean, it's not intact, but this envelope gene has been co-opted and is now a human gene called, some people say syncytin or syncytin. I'm not sure what's technically proper, but the reason why we are placental mammals is because of some ancient viral infection. Fascinating, yeah.
yeah. Well, that makes sense. There's all this work on the microbiome, right? So it's pretty clear we have symbiotic relationship with, in this case, whole bacterial cells. Why not symbiotic relationship with,
Yeah.
viral remnants or and so on? Yeah.
Cool, yeah.
Uh, and then, so you already mentioned the reverse transcriptase inhibitors. Have you thought, well, I'm sure you have, more about the implications of your work for prevention and treatment? What are you doing in the flies? You've got these models now.
Yeah.
You can reliably look at the pathogenic processes in cell, the transfer between cells. Are you doing manipulations?
Yeah, I mean, we want to know how they transfer between cells. Is there a cellular receptor for that? We want to know whether or not the virus transferring actually moves the aggregated protein or whether its movement triggers aggregation in the recipient cell. We're very interested in what's toxic about these retroviruses or
Yeah.
retrotransposons. I mean, it could be the DNA damage that they cause. It could be the sort of triggering the innate immune system for leading to sort of inflammatory responses. So that's something we're very interested in as well. Um, and then we're interested in terms of what's toxic. I think there's clinical implications because, if we're right and these are contributing to the pathology in some way, then it's very important to understand what step of the virus's life cycle or attempted
Yeah.
replication cycle is it really triggering cell death and spread of that cell death? Because if it requires reverse transcription, then reverse transcriptase inhibitors would be wonderful drugs to try. If it's just the RNA that's toxic, then we might want to target other pathways. Could be both,
Yeah.
could be neither. So we're really trying to dig into that basic biological question, even in human cell culture, with the human endogenous retrovirus. How does it trigger the aggregation of the TDP43 protein?
Yeah.
What aspect of the viral life cycle triggers that feedback amplification?
Now there are high throughput screens. When I was at NIH, they have this facility down in Rockville where if you have a 96 well plate fluorescent assay, they have 200 and some thousand chemicals.
Yeah, yeah.
And are there what's going on there? I mean, can you tap into that? And
I mean, it's a really great suggestion. My feeling about high throughput screens is you need a really good assay. You
That's
need to
right.
be
Yeah.
sure that, first of all, the assay has to be robust and it has to be one that's amenable to high throughput,
Yeah.
like a
Yeah.
fluorescent reporter or something like that. Um, and you also want to be sure that the biology you're assaying is the relevant one. So personally, I feel like it's not quite time. I mean, some people say, cowboys just get on horses and they hang on until the horse throws them off. And like, there's something to be said for that. I personally feel like before I would sort of change my approach to really get involved with high throughput screening, I really want to know, do we have good evidence? Do we have
Yeah.
solid evidence that the thing that we're
Yeah.
assaying is the right thing? And
Yeah.
there's that old saying, don't mistake my enthusiasm for my idea with confidence that I'm right. Like I'm really enthusiastic about all my ideas, but I fully admit that, until we really nail down mechanisms and show them to be at play in humans, any idea that you come up with could be wrong. It's still a good idea to investigate it, but could turn out to be wrong.
Yeah. Okay. Do you have any parting comments before we wrap it up?
I don't really. I mean, it was a
Yeah.
really wide-ranging and wonderful interview and discussion. I watched a few of your podcasts in preparation for this, and I've noticed that they tend to be really like wonderful. Open. It's almost more like a discussion between
Yeah.
scientists.
Yeah.
Yeah,
Yeah.
yeah,
It's not really an interview. It's a talking back and forth. And
exactly. Yeah, yeah,
yeah, I enjoy doing this since I retired from NIH. I have to have something to do in the morning, so
yeah,
I do this, and that lets me keep my brain thinking about neuroscience, right?
yeah. Absolutely. Are those horse bits behind you?
They are my grandfather, my father, me, my brother trained and raised harness horses,
Oh,
trotters and
wow.
pacers.
Yeah, cool.
Actually, they have tracks up in New York, Yonkers, Meadowlands.
Oh, the
Meadowlands
Meadowlands. Yeah.
now is like the biggest, harness horse racing place in the country.
Oh, really interesting.
And yeah, these bits are so for the harness horses, you have a driving bit, a bit that goes in the mouth, like this one. It's attached to the lines, and then the smaller one goes in the mouth, and it's called a check bit. And essentially, it's used so they keep their head up when they're going. And those are bits from our two best horses. This is a long time ago now. This is
Yeah.
in the 60s, 60s and 70s when I
Wow.
was,
Yeah.
and I drove myself in races. Uh, yeah.
Amazing. Yeah.
I
I've
used
seen
to
that. It's
do,
an amazing sport.
it is. It's a lot of, we didn't do it for a living. It was a hobby. We lived on 100 acres in southern Minnesota. We had a training track. And actually, my father was a prosecuting attorney for Olmsted County
Oh, wow.
in Minnesota. And our neighbors farmed our land and then we split the crops 50-50. So we had harness horses and we had beef cattle. And yeah, so I grew up, that's why I asked you where you grew up. So insects in a city, insects are one thing that there are insects around. Uh, but it's actually, I don't know if you've noticed this, and I've seen a dramatic decrease in overall in insects over the last 30 years.
Definitely.
It's dramatic.
Definitely. You
You know,
remember
we used,
driving cross country and your windshield would be just,
yeah,
your grill would just be
yeah.
covered. Yeah.
Yeah, and you go if you have an outdoor light on your house, it'd be surrounded by moths and
Right.
other flies at night. Now I go out here at night and
There's almost
I
nothing.
think this is a huge problem, the decrease in biodiversity.
Absolutely. Yeah. I mean, they're really important biomass for
So,
the
but
whole
you're
ecosystem.
keeping the fly population up, so that's good.
There's no problem with the laboratory fruit fly population. Yeah.
Okay, thanks a lot, Josh. I enjoyed
You're
this.
welcome. I really enjoyed it too. Yeah. Take care.
You too. Bye.
Bye.